getting used to the hybrid thing. After the hour. Okay. That is an MIT. Give students go 4 minutes before and the next class after. It's pretty big. They can go from one class to another. So it tends to talks. I'm afraid people automatically use it. All right. Well, good afternoon. So I'm standing in for Dara Manoach, who unfortunately could not be here today. But she sends her apologies, and she's on the YouTube channel listening in remotely. So Dara said she met Sundari about 2 years ago in the midst of COVID, right after Sundari was recruited to MGH, and she was looking forward to hearing and meeting Sundari, but she's online. So Dr. Sundari Chetty is an assistant professor at the Center for Regenerative Medicine at MGH and the Department of Psychiatry at Harvard Medical School. And she also serves as a principal faculty member in the Harvard Stem Cell Institute. So Sundari received her Ph.D. in neuroscience and her BA in molecular and cellular biology from UC Berkeley. She did a postdoctoral fellowship with Doug Melton at Harvard, and then soon after, in 2016, she started her lab at Stanford University. And in June 2022, she relocated her lab to MGH. So the goal of Sundari's lab is to understand the mechanisms underlying neurodevelopmental and psychiatric disorders such as ASD and schizophrenia, and identify novel therapeutic targets. Her lab has been investigating the mechanisms underlying brain overgrowth or undergrowth using human-induced pluripotent stem cell technology, using human iPSCs from people with idiopathic neurodevelopmental disorders as well as those with known genetic variations. Her lab has used two-dimensional and brain organoid models to investigate changes at the cellular, functional, and mechanistic levels using a wide range of techniques. Her elegant work has been published in high-impact journals. This includes a study of brain overgrowth in 16p11.2 deletion syndrome, which some of us have also studied as part of the Simon Center for the Social Brains Targeted Project. This work was published in PNAS and was hailed as a scientific breakthrough in revealing cellular mechanisms of ASD and pointing to novel therapeutic avenues in an accompanying editorial. So, welcome to the Simon Center, and we look forward to hearing your talk.
Well, thank you very much for that kind introduction. And thank you to Dara and Morganka for hosting me and inviting me to the Simon Center. It's been a joy to meet all of you today and get to learn more about your work and form new collaborations. So as Morganka mentioned, my lab's using human pluripotent stem cell models to understand neurodevelopmental and psychiatric disorders. One of the main neurodevelopmental disorders that we've been focusing on is autism spectrum disorder. And one way that we've been focusing on autism spectrum disorder is that given that it's such a heterogeneous disorder, we've been trying to identify clinically meaningful subtypes that we could model using human iPSCs. One specific subtype that we've been focusing on is brain overgrowth or megalencephaly that occurs in about 20% of autistic children who have disproportionate megalencephaly. So relative to the autistic kids who have a normal brain size, those within large brains tend to have more severe behavioral and cognitive deficits. They have lower gains in IQ over time. They are also less responsive to standard therapeutics. They have lower language capabilities. So on the spectrum, they tend to be much more severe relative to autistic kids who have a normal brain size. And so we've been trying to understand the molecular and cellular mechanisms leading to brain overgrowth in hopes that we can identify novel therapeutics that might help alleviate some of these symptoms. So neuroimaging studies have shown that the increase in brain size precedes the first clinical signs of autism. MRI scans have shown that there are increases in both gray and white matter suggesting that there are changes at the neural level as well as at the oligodendrocyte level. And so within my lab, we've been using human iPSCs to model this megalencephalic phenotype in a number of neurodevelopmental and psychiatric disorders. So one of the projects that we began at Stanford was looking at 16p11.2 copy number variation, including patients who have the deletion syndrome and who have autistic-like symptoms, and trying to understand how brain overgrowth occurs in those patients. Another project, which I'll touch upon later in the talk, is in collaboration with colleagues at UC Davis at the Mind Institute with David Amaral, where we have generated about 40 human iPSC lines from typically developing and autistic kids who either have a normal brain size or the disproportionate megalencephaly, and they don't have any known genetic causes of ASD. And so these are more idiopathic forms of ASD. And our hope has been to try to use these models to better understand brain development in neurodevelopmental and psychiatric disorders. So the main areas that we've been focusing on in the lab is looking at whether there are changes in cellular proliferation, cell survival, cell size, and or the elimination of cells through the neuroimmune system. So in the first part of the talk, I'll focus on our work looking at 16p11.2, primarily the deletion syndrome that's associated with a macrocephalic phenotype and autistic-like symptoms in comparison to those with 16p duplication syndrome, which can have a microcephalic phenotype, but in our study we used patients who have a normal head size. And the main focus of this study was to look at mechanisms that were contributing to brain overgrowth. We decided to focus on CD47, which is commonly known as a don't eat me signal. It's commonly upregulated in a lot of cancer and tumorigenic cells, leading to the cancer cells avoiding engulfment by microglia or macrophages. So we were very interested in looking at whether these types of mechanisms are at play in neurodevelopmental disorders where there may be overproliferation of cells contributing to the brain enlargement. So we first used human iPSCs that were derived from patients with the 16p11.2 deletion or duplication. And here you can see that about 29 genes are affected in this syndrome. And so we obtained iPSCs through the Simons Foundation. The patients with the 16p deletion syndrome had an enlarged head circumference, so above the 97th percentile. And those in the duplication patients had a normal head circumference of about 50th percentile. And we also had some control iPSC lines from typically developing individuals. So we first confirmed that genes, at least a subset of the genes within the 16p locus, are either suppressed in the deletion syndrome or upregulated in the duplication syndrome, as you can see here. We next wanted to model gray matter, and we specifically were looking at progenitor cells in this project, where there may be changes in cell proliferation and mechanisms associated with cancer. And so we used a two-dimensional directed differentiation protocol to differentiate the iPSCs into neural progenitor cells. These neural progenitor cells following the differentiation express a number of neural markers, such as PAX6 at the mRNA and protein level, and you can see comparable expression across the different patient groups. We next looked at whether CD47 would be overexpressed, similar to cancer cells. And we saw that indeed in 16p deletion NPCs, there's an increase in CD47 expression at the mRNA level. and at the protein level as well, using flow cytometry to look at cell surface expression of CD47. We wanted to look at this in white matter as well, and to model white matter cells, we used a 50-day directed differentiation protocol to generate oligodendrocyte progenitor cells. And we looked at CD47 expression at an early time point of about 12 days and saw no significant changes at an early time point when the cells have not fully matured into an OPC state yet. However, after 50 days, there was a significant increase in CD47 expression in the 16p deletion conditions. Here you can see the expression of O4, a cell surface marker of oligodendrocytes, and increase in CD47 also at the protein level. Calreticulin is an eat me signal that's often upregulated on cancerous cells. This indicates that the cells are unhealthy in some way and therefore should be eliminated. We wanted to see if our cells are also overexpressing calreticulin and potentially may indicate that they're unhealthy. And we saw that in the 16p deletion condition, both NPCs and OPCs have an increased expression of calreticulin. Calreticulin binds to a lectin on the cell surface known as PHAL. That lectin is also upregulated in both NPCs and OPCs, specifically in the deletion condition where there's brain overgrowth. And so this indicates that the cells should be eaten or eliminated. However, CD47 is typically the predominant signal, and therefore they may not get eliminated. So we next wanted to investigate whether this actually translates to any changes in cellular phagocytosis. So we co-cultured our NPCs with human-derived macrophages and looked at changes in cellular phagocytosis 2 hours later using fluorescence cytometry. So what you can see here is that suppressed phagocytosis in the CD45 positive NPC population in the 16p deletion condition where there's overexpression of CD47, suggesting that the cells are not getting eliminated and this may be contributing to the brain overgrowth. There are several blocking antibodies that have been developed for cancer to block CD47, so we wanted to test if that would help revert the changes in phagocytosis. So we treated our cells with CD47 blocking antibody and then performed the phagocytosis assays. And in this in vitro experiment, you can see that there's a restoration of those rates to normal levels. And similarly in OPCs, after 50 days of in vitro differentiation, we added a cell trace dye to the OPCs and co-cultured them with macrophages and did the phagocytosis experiment. And here you can see in the deletion condition, there's suppression of phagocytosis that's restored if we pre-treat the cells with a CD47 blocking antibody. Next, we wanted to see whether these changes would translate in an in vivo setting as well. So we, after the 12 days of in vitro differentiation or 50 days for the OPCs, we intracerebrally injected the iPSC-derived cells into NSG pup brains and looked at phagocytosis at an acute stage 24 hours later, along with markers CD11b and CD45. And you can see in the deletion condition, there's significantly less phagocytosis of the NPC population. But if we treat with the CD47 blocking antibody prior to the transplantation, we're able to revert those rates of phagocytosis to control levels. And similarly in OPCs, after 50 days of differentiation, there's a suppression of phagocytosis in the deletion condition where there's brain overgrowth that can be restored to control levels following treatment with the CD47 blocking antibody. So next we wanted to look at whether we could administer the CD47 blocking antibody systemically, which would be more therapeutically relevant. And in cancer, it's been shown to cross the blood-brain barrier. So we used a different CD47 blocking antibody here that's been in clinical trials, HU59G4, and systemically administered it and transplanted the NPCs or the OPCs into the pup brains. What you can see here is that in the presence of the blocking antibody, there's increased rates of phagocytosis in the deletion NPCs, and similarly in the OPCs as well. In immunosectioning, you can see here the STEM 121 marks the human cells, and in the deletion condition, in the presence of the blocking antibody, you see increased rates of phagocytosis with IBA1 positive cells co-localizing with the human cells that have been transplanted. So next we wondered, is this specific to the patients who had the macrocephalic phenotype? And so we added some more iPSC-derived cell lines from the deletion patients who have now a normal head circumference of about 50th percentile, and then differentiated them into NPCs or OPCs. Following differentiation into NPCs, we did not see the same increase in CD47 expression or calreticulin expression as we did in the patients who had the brain enlargement. And similarly, in OPCs, after 50 days of directed differentiation, we didn't see the same increase in CD47 expression or calreticulin in the patients that have a normal head circumference. And so this was suggesting to us that it's particular to the brain overgrowth patients with the deletion syndrome. And strikingly, anti-CD47 treatment did not increase rates of phagocytosis in the NPCs or OPCs from these individuals who have a normal head circumference. And so the increase in calreticulin is important for the blocking antibody to target those cells. So what this part of the work has enabled us to show is that megalencephaly or macrocephalic phenotypes can be modeled using human iPSCs to better understand the molecular and cellular mechanisms, and that pathways disrupted in cancer, such as CD47, may be disrupted in neurodevelopmental disorders where there is brain enlargement. And that the balance between these eat me and don't eat me signals may play critical roles during neurodevelopment. One of the ongoing work in our lab is to look at the other don't eat me signals in these neural cells where there's brain overgrowth. And some of the other ongoing work related to the 16P work is to look at CD47 isoform expressions, and we see that particular isoforms are overexpressed in the NPCs and OPCs where there's brain enlargement. And we would like to look at CD47 expression in postmortem brain tissue where there's an autistic phenotype associated with the megalencephaly. That's been a little bit harder to identify because a lot of the brain banks don't have the information tagged with it, whether it's from a megalencephalic phenotype or not. And investigate which of the 29 genes within the 16P locus directly regulate CD47 pathway and calreticulin, and this could have implications not only for neurodevelopment, but for cancer as well. And in the next part of my talk, I'll focus more on an NICHD-funded work, an Autism Centers of Excellence grant that we got in collaboration with David Amaral and his colleagues at the UC Davis Mind Institute. So in this project, as I mentioned earlier, we are focusing more on the mechanisms regulating brain overgrowth in idiopathic forms of autism spectrum disorder. And so in collaboration with David Amaral's group, we have generated about 40 induced pluribone stem cell lines from typically developing kids or autistic kids who have a normal brain size or with a disproportionate megalencephaly. And the idea is to differentiate those iPSC lines into neural progenitor cells, oligodendrocyte progenitor cells, and microglia to understand the mechanisms leading to brain overgrowth, and correlate our cellular findings back with behavioral data and neuroimaging data on the same children where we've derived the iPSCs. And the hope is to identify novel therapeutics in a more patient-specific manner. So in this project, we had our goal is to generate 40 iPSC lines, 10 each from autism spectrum disorder with disproportionate megalencephaly, autistic children with the normal brain size, typically developing kids with megalencephaly, and typically developing kids with the normal brain size, so that we can better understand mechanisms leading to brain overgrowth and then those that are specific to autism with brain overgrowth. While we were recruiting subjects into this project, we also initiated a study from pre-existing cohorts of children who have been part of the UC Davis Mind Institute Autism Phenome Project. And these children have been longitudinally tracked from the ages of 2 to at least 12 years of age. And they have neuroimaging data, cognitive, behavioral, and medical data. So we generated induced pluripotent stem cells from at least 12 of these children, from typically developing kids with the normal brain size, autistic kids with the normal brain size, and autistic kids with the disproportionate megalencephaly. And all of these lines have been differentiated into neural progenitor cells using a two-dimensional protocol. And we then performed RNA sequencing on the NPCs to better understand the genes and pathways that may be deregulated. So this includes all the 12 subjects that we generated iPSCs on, and you can see that the ADOS severity score is similar in the ASDN and ASD-DM group, but as you'll see, some of the mechanisms are very different. This again shows that the ADOS severity is similar between the two autistic cohorts over time, but other clinical and behavioral characteristics can vary over time. So you can see here, verbal ability is much lower in the ASD-DM group over time, even out to about 11, 12 years of age. And then similarly, social communication and other behavioral measurements are also lower in the ASD-DM group over time, even out to about 12 years of age. So what we did was we took PBMCs from all of these individuals, reprogrammed them into induced pluripotent stem cells, and then directly differentiated them into neural progenitor cells over a 12-day period. These cells express many of the pro-neural markers, including SOX1, nestin, at comparable levels across the different patient groups, and also other markers such as PAX6, both at the mRNA and the protein level. So we then performed RNA sequencing on these NPCs and did principal component analysis. Here you can see that there's a nice segregation of the populations of cells where TDN separates out from the ASDN and the ASD-DM subgroups. And then looking at the genes that are differentially regulated, we find about 200 to 200 genes that are significantly up or down-regulated. And gene ontology analysis has shown that a lot of these pathways are associated with cell proliferation and cell-to-cell communication. We also did weighted gene co-expression network analysis, WGCNA, to look at how genes and modules are differentially regulated. One of the modules that is specifically upregulated in the autistic condition with the disproportionate megalencephaly includes many pathways associated with cell cycle, cell division, and brain development. So this enabled us to then look at cell proliferation further. So we did a number of assays to look at changes in cell proliferation, and MTT metabolic assay to look at cells that are more viable in the different conditions, looked at cell doubling time to see how quickly a cell culture doubles, and BRDU incorporation to see if they're replicating at a faster rate. So what you can see here is that in autism with a disproportionate megalencephaly, there's increased cell viability and also a decrease in the cell doubling time, indicating more proliferative cells. And here it's confirmed by the BRDU incorporation. So indicating that the ASD-DM and PCs are indeed more proliferative. Modules that were unique to the autism with the normal brain but suppressed in the DM included a lot of positive regulation of programmed cell death and P53 signaling pathway, and therefore we decided to look more into cell death pathways, potentially indicating whether in ASD-DM perhaps the cells are not dying and they might be stressed. And so we looked at cell stress and cell death related pathways. Specifically, we looked at a number of apoptotic proteins, including cleaved PARP1, which indicates less apoptosis in the ASD-DM condition, and cleaved caspase-3, you can see, is also suppressed in the ASD-DM. If we do an ELISA for a cleaved caspase-3, that was also significantly downregulated in the ASD-DM and PCs. And here you can see RNA sequencing results, transcripts per million for caspase-3, also indicating a downward trend in the DM and PCs, indicating less apoptosis of the megalencephalic NPCs. So we also looked at another programmed cell death pathway known as ferroptosis, which is also commonly implicated in cancer. Some of the molecular and cellular features associated with ferroptosis include smaller mitochondria or dysfunctional mitochondria, iron accumulation, and lipid peroxidation, including oxidative stress. And one of the key regulatory genes associated with this pathway is GPX4. And so we decided to look at this further as well. We looked at ferrous iron accumulation in the NPCs, and we saw that in the DM NPCs there is a significant increase in ferrous iron accumulation. In many neurodegenerative disorders, it's been shown that an accumulation of this can be very toxic to the neural cells. We also looked at cellular stress and oxidative stress. And this is a measurement of the reactive oxygen species, and you can see an increase in the DM NPCs. Another way to look at oxidative stress is to look at the ratio of GSH to GSSG ratio, glutathione, and here you can see a significant decrease in the DM condition, indicating more oxidative stress and that the cells are in an unhealthy state. We also used a human cell stress array to look at a number of cell stress-related proteins. Some of the main proteins that seem to be upregulated in ASD-DM include the phospho-heat shock protein 27, as well as a few others such as CA9, and both of these have been shown to be overexpressed in many types of cancer and associated with stress-induced survival of cancer cells. And strikingly, GPX4, which is an inhibitor of ferroptosis, is upregulated in the ASD-DM condition. So we think there's increased oxidative stress but a reduction in cell death-mediated pathways through apoptosis and ferroptosis. We've also looked at mitochondrial function. This is preliminary data where we're replicating it using the seahorse assay. And what you can see here is that there's a change in the glycolytic rate of the ASD-M NPCs relative to the TDN and ASDN NPCs. However, mitochondrial activity is suppressed in both ASDN and ASD-DM, as shown by the oxygen consumption rate. And we have some preliminary data looking at also electron microscopy images of the mitochondria in the NPCs, and we see smaller mitochondria in the ASD-DM NPCs that we're replicating currently. So what this work has been showing us currently is that there's increased cell proliferation and suppression of many cell death-mediated mechanisms. The hallmark proteins that are associated with apoptosis and ferroptosis are suppressed in ASD-DM, and that the ASD-DM NPCs show increased oxidative stress through increased production of reactive oxygen species and other cell stress-related proteins, as well as the increased iron accumulation. Despite this increased cellular stress and iron accumulation, these NPCs are upregulating pro-survival mechanisms and evading cell death. So we currently see an upregulation of GPX4, an anti-ferroptotic marker, and increased in phosphorylation of heat shock protein 27, which has been associated with stress-induced survival of cancer cells. And so we're now exploring whether a lot of the agents that have been developed for cancer, whether we could use those here to target the ASD-DM NPCs. And current work, we're also looking at whether any of the cellular findings could be correlated back with in plasma samples or whole blood samples, including metabolomics data on these same individuals. And we have this data on about 300 to 400 of the participants in the Autism Phenome Project. So we are currently looking at whether any of these cellular markers could potentially serve as a biomarker in blood samples or other samples that we have. And in neurodegenerative disorders, it's been shown that iron neuroimaging to look at iron deposits in the brain is a predictor of many aging-related disorders. And given our cellular findings of the increased iron accumulation, we are thinking whether this could be a potential means to look at, to potentially even identify those who might have ASD-DM at an early stage. And ultimately, what we hope to do is identify the signaling mechanisms regulating brain development in neurodevelopmental as well as psychiatric disorders. As I mentioned, we're very interested in correlating this cellular data back with behavioral data or brain imaging data on the same individuals. And ideally, we will also correlate and validate these findings in postmortem brain samples from patients who have the macrocephalic phenotype. And the hope is to identify novel therapeutics such as CD47 treatment or others, such as those targeting the apoptosis or ferroptosis pathways, to help mitigate some of the symptoms that are associated with brain overgrowth and autism. So with that, I'll end. A lot of this work was initially initiated at Stanford by many of my former postdoc members, including Jingling Li, Thomas Brickler, Anna Sherbana, and Liang Ma, and all of our collaborators at the UC Davis Mind Institute and at Stanford. And the lab over the last year has moved to MGH and Harvard Med School. And a lot of the newer work, especially the idiopathic NPC work, was done by a postdoctoral fellow in my lab, Shui Chen, alongside several of our other postdocs, including Janani Ramesh, Mamoon, and our research technicians. So thank you very much, and I'm looking forward to getting your feedback, too, on our work.
we were talking earlier, on the one-on-one, and I was sort of thinking about like the overgrowth where there's some normalization, right? And
Right.
And thinking, well, what the sort of the more persistent phenotype
Yeah,
might be.
right.
And you might think that like the oligodendrocytic,
Yeah,
proliferation effects might correlate in some way with like hypomyelination. And so
Right.
just think about the last part. Is there any way to look at the oligodendrocytic,
yeah.
the OPCs? Is there any kind of selective vulnerability between the NPCs and OPCs that might allow you to see whether
Yeah, that's a good
it's like
question.
like a specific targeting, maybe therapeutically, of
Right.
OPCs that might continue to show the overgrowth phenotype and then lead to this.
Right, right. Yeah, with the idiopathic lines, we haven't looked at the OPCs yet. We haven't differentiated them into OPCs, but it's something that we plan to do in the near future. And then we could do some co-culture experiments as well, where we could see if the oligodendrocytes are secreting anything that may affect the neurons. The one work that I didn't get a chance to touch upon is we've differentiated about 20 of these iPSC lines into microglia, and we are seeing much more activated microglia in the autistic conditions and more sustained activity in the ASD-DM, so microglia are phagocytic throughout time in the ASD-DM conditions, and in the ASDN condition, it's much more acute phagocytic activity. And it's significantly more relative to the typically developing microglia and also relative to a primary microglia cell line.
So there are about 25 people online, but Dara had written some questions. She's really sorry to be here. But I have the same question, which is that when I look at the plot early in your talk of the 16-peak deletion individuals, there is a wide range of head size or macrocephaly. And so is that due to protective mechanisms, you think, or is that due to some other factor? Why the variability?
Right. So in our case, we used patients above the 97th percentile, but you're correct that in the syndrome, there's a wide variety of individuals. And I think that has to do with, which genes are affected. I think it's such a heterogeneous disorder, and there's a different dosage effect leading to the brain enlargement or not.
And the duplication
Yeah.
has much less severe phenotype. And what are your thoughts on that? It's not a symmetric
Yeah.
situation with respect to CD47-based phagocytosis, you think?
Yeah, so we have looked at 16p duplication individuals with a very small brain, microcephalic phenotype less than third percentile, and head circumference. There we are seeing more cellular elimination. And so we do see the opposite mechanisms on the extreme ends, but those who have the normal head circumference, we don't quite understand the neural mechanisms leading to any phenotypes they are experiencing. Yeah.
Great talk.
Thank you.
I have two questions. The first being related to the second half of the talk. Have you continued the OPC differentiation into neurons to see if they're also expressing increased cellular stress?
That's a good question. We haven't yet. We have done that with the 16p lines, where we do see the increase in CD47 expression, even with further differentiation into cortical neurons. But we haven't looked at it in the cellular stress idiopathic forms of autism yet. Yeah.
Kind of related to variability in patient phenotypes, are you looking at any polygenic risk scores for your cell lines to see if those are different across your conditions?
We haven't, not yet. Yeah.
I have another question. So one of the other mechanisms, as we were discussing briefly when we were chatting, for larger brain size is P10 mutation,
Right.
which upregulate PI3K, KT
Right.
signaling, and that's a very well-known mechanism for growth and
Right.
development in all cells, including neurons.
Yeah.
So is your mechanism completely independent of P10? Is there some interaction between P10 signaling and CD47 signals?
It's a good question. We haven't looked at it yet. P10 mutation is also associated with autism. And we haven't generated iPSC lines on, we have been thinking about it, but haven't generated iPSC lines on those individuals to look at whether the mechanisms would be similar and how CD47 would play a role into that. Yeah.
And yeah, Ravi.
That was really elegant mechanistic dissection. I have kind of a naive question because the goal of so much research, especially in like the stress space and aging space, is like promoting neurogenesis and promoting neuronal survival. And even in infancy, we think of the brain as super plastic. Like infants can recover dramatically after neonatal stroke or neonatal meningitis. So I feel like the brain should have ability to adapt for increased survival of cells. Like,
Right.
so what else do you think is going on in terms of the mechanism for why even typically developed megaloencephalic kids have seizures and other problems?
Yeah.
Why can the brain be plastic when there's injury and increased neurogenesis to compensate, but not at baseline in a healthy state?
Yeah, it's a good question. I don't know if I have an answer for that, but I think maybe just the additional plasticity going on early in neurodevelopment and the pruning process that's occurring. I think that's playing a critical role, whether it's typically developing brain or the diseased brain. So, for instance, Beth Stevens' lab has shown that early in neurodevelopment, CD47 is playing an important role in just normal synaptic pruning. And so I think even in the normal cellular homeostasis that's ongoing in normal neurodevelopment, I think these eat me, don't eat me signals are playing important roles.
Thanks.
Do you think therapeutics for that target CD47 are feasible in the context of brain development, given that they target cancer mechanisms? How do you think of CD47 based therapeutics to be applicable in
Yeah, so what we hope to do is try anti-CD47 treatment in a mouse or another animal model where there's neurodevelopmental defects associated with the brain overgrowth, and see if that helps mitigate any of the cognitive or behavioral symptoms. I think the one advantage is that the blocking antibody targets the calreticulin positive cells and not the healthy cells that are not overexpressing calreticulin. So it at least provides a mechanism to help eliminate those cells and see if that actually leads to changes in behavioral and cognitive outcomes. To at least test it and see if the change in brain size leads
So
to...
on that note, you think that actually targeting brain overgrowth or numbers of cells would be
sufficient.
effective as opposed to some,
Yeah.
we physiologists
Right.
tend to think of
Yeah.
circuits and
Yeah.
cell types
Yeah.
and the information processing
Right.
carried out by these mechanisms as fundamental for cognition.
Right.
How should we think of cell size as
Yeah.
being, or brain size as being causal for these
Yeah.
kinds of capacities?
I think it's possible that these cells that should have been eliminated are harming the other
Right.
normal healthy cells in some way, and therefore removing them can help clear out that debris in there and
What kind of evidence would we need to show that actually the cells that survive are actually contributing to dysfunction?
Yeah, I mean, it's a good question. We have a lot more work to do to figure that out.
Because on the flip side, I've been amazed by these essentially anecdotal studies of you have hydrocephalus and the brain is so thin and
Right.
there are probably fewer cells.
Yeah.
And the person was normal and this was randomly discovered as a result of a brain scan
Right.
for something else.
Right, right.
And stuff like that. So the
Yeah.
brain, as Ravi pointed out, has
Yeah.
enormous
Yeah.
potential for compensation or for rewiring, at least in the face of major changes,
Right.
at least
Yeah,
in childhood.
yeah, yeah.
But you don't necessarily know that it's even the gray matter,
Right.
proliferation. I mean, again, it could just be the oligodepressive
Yes,
effects.
that is
might be
possible.
little
Yeah.
regular and
That-
stuff that
that
are
is-
not necessarily contributing to phenotype.
Indeed.
Yeah. Yeah, one thing that we're seeing in the iPSC-derived microglia is that they're secreting more pro-inflammatory cytokines in the autistic conditions. So we've collected the condition medium and we'll be applying it to neural cultures to see if that also has an effect on the neural side of things.
Related to that point about brain overgrowth in the number of cells, do you think that because there's more growth or more cells and more divisions that those neurons have a mutation?
No, that's a good point. We haven't looked at that yet. Yeah. Exactly, right.
All right. Any other questions for Sundari? Comments? Well, in that case, let's give it another.
Thank you. Thank you very much. Yeah, right. Yeah.
That's the oligodendrocyte. So you think there is more myelin as well? Or not necessarily?
I think so. The neuroimaging scans are showing increase in white matter.